Okay, hi, hello, welcome to the Physionic Podcast, which is more of a lecture again for the second week in a row. If you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a doctorate candidate in molecular medicine, hold my master's degree in exercise physiology. I worked in a muscle metabolism laboratory for quite a while, and now I'm working in a cell biology lab as I go through my PhD in medicine. So, what are we going to be talking about today? We will be discussing the second part of a muscle lecture that I gave last week. It was actually the lecture that I gave to my undergraduate class, and I thought, why not share that information with the physionic community? So I never think that these lectures are actually going to get, many views or anything of that nature, but I was pleasantly surprised that the last lecture, which was about an hour long, actually wasn't that far off. It gained a decent amount of views, but really I'd like to quick thank, before I actually get into this lecture, I'd like to thank everyone that watched or listened to the podcast before that was discussing metabolism on fat loss and reconciling calories with hormones. I got so many great comments. Some people that pushed back, just some great feedback, and I really appreciate it. And then I had this warning at the beginning of, you have to watch this entire thing. Like, it is the most important podcast. And it is. I still think it is the most important podcast I've ever released. 2 weeks later, who would have thought? But really, I said, watch the whole thing. And then I said, I can check in the analytics if people actually watch the whole thing. And on average, it was much higher for a really long video like that. It was much higher that people watch the whole thing. So if you're one of those people, you rock. You're awesome. That is true drive for education, for knowledge. So pat yourself on the back. That was awesome. Okay, I wanted to say that last podcast, but I forgot. Okay, now this podcast is going to be discussing the second part of muscle physiology, muscle lecture, and I'm going to be covering muscle metabolism in this lecture, and then I'm going to do a little bit of a recap on last lecture, because the reason why is, if you are brand new and you haven't watched the first lecture, it's like an hour long, so it's quite long. If you want to know all the details, of course, start there, and then that will kind of give you the groundwork up to this point. But I'm going to give you a quick recap in case you just don't want to go through everything else and you just want to learn about muscle metabolism, because at the end, I'm going to have a mystery. I'm going to have a clinical scenario that we're going to solve together. And I'm going to give you enough pauses within that scenario so you can kind of think about it. And then I'll give you the answer at the end, and I'll explain the answer. So if you're interested in riddles and whatnot, and you don't need to know any other prior information, just what was in the last lecture and what you're going to be learning in this lecture, which you'll be getting that recap right now. So let's go ahead and jump into it. All right. So this is what I did with my undergrad students. I told them to kind of piece all of these parts together. Now, if you watch the last lecture, you might be able to piece all this stuff together. And it's really just kind of going in order of these different actions that occur on the muscle, in the muscle, and around the muscle. And I'm just going to give you the answer here, because I'm actually going to show you what this actually looks like. But first, you have the neuron action potential, so that electrical signal that starts in the neuron, which is the nerve cell, which then releases acetylcholine, number 2 here, and then that switches on, that activates through voltage-gated sensors, or really, just like the neuron was activated through an action potential, the muscle is then activated through an action potential, which then leads to this activation of the dihydropyridine receptor, which then mechanically activates the ryanodine receptor. Why that was important is because the activation of these two receptors leads to the release of calcium from the sarcoplasmic reticulum. And don't worry, I'm going to show you all this if you haven't, if you didn't go through the first lecture in just the next slide. And then there's the interaction because of the calcium release. There's the allowance for myosin to then interact with actin. So let's look at that. Okay, so here we are at the first part, and we've got our neuron here, which is kind of a big picture here. So we've got our neuron. So the end of the neuron is called the axon terminal, and here we've got our muscle cell. And so this is really granular, right? We're not looking at, entire tissues. We're looking within tissues at single cells and even just compartments of single cells. So our neuron has an action potential that if we zoom in, our neuron has an action potential that comes down, which is an electrical signal. And what that looks like, if you were to actually read it on a graph, is you have the inside of your cells are negatively charged. So there's a negative potential or a negative gradient between sodium, which is outside of the cells, and potassium, which is inside the cells. And this is a really basic explanation, but let's just go with it because it works well. So there's negativity inside the cells, and there's more positivity, quote unquote, positivity outside of the cells. So this action potential has this negative, 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 and then suddenly when it gets activated, the actual action potential itself is a sudden rise in positivity as the things that are outside of the cell will enter the cell. And this leads to a more positive potential. Now, ultimately, that goes all the way down the neuron and eventually has to stop because the neuron stops. It doesn't go beyond its own existence. So it eventually, the action potential will then die down and go back down to negative, and this is called a repolarization, and then a hyperpolarization, and then come back to its normal resting potential, which is negative. But that whole sequence of things ultimately triggers this release of a chemical, of a particular molecule known as acetylcholine. And acetylcholine, which is found in vesicles or little sacs inside this axon terminal, will then be fused with the membrane and be released. So acetylcholine is then released into the space between the muscle cell here, and the neuron. So this space here is called the synaptic cleft. And here, with all this acetylcholine that's now been released into the synaptic cleft, acetylcholine will then bind to the acetylcholine receptors, which makes sense. And this will then, the same thing that I described here with this sudden rise in potential, will happen in the muscle cell, as the receptor will go from a closed, what's known as a closed conformation, or the way that it looks, the structure is closed. Once acetylcholine binds, it opens it so that the sodium can then, which is positive, come into the cell and make this whole muscle cell positive. So this then occurs inside the muscle cell. And that's why I've got all these positive icons. Okay, so then let's look at that. Here's everything that I described, and just a little bit off to the side, and we've got all this sodium. It's just popping off all these channels. All these voltage-gated channels are just opening up, and more sodium is coming in, and it's just propagating all the way down the muscle cell, and then it's coming down, and then it's activating this dihydropyridine receptor, which then is mechanically linked. So there's an actual physical link between the dihydropyridine receptor and the ryanodine receptor. And this link opens up the ryanodine receptor, and calcium then floods out of this compartment known as the sarcoplasmic reticulum. It's the same in other cells as known as the endoplasmic reticulum. So this compartment then releases calcium, and then we get an active muscle cell. And that's really how muscle contraction occurs. So another thing that I covered was the types of muscles. So there's type 1 and type 2 muscle cells. And one of the great ways to distinguish this is actually to take a sample of muscle cells and to then slice pieces of it so that you can actually look down the tubes. So muscle cells look like tubes. They're very long. So what researchers do, what you can do is take a biopsy take a punch biopsy. So you're just taking a small amount of muscle, about 50 milligrams, something like that, 100 milligrams. And then from there, you end up slicing really, really thin slices. Think of it like going to the butcher shop. And if you've ever watched The Butcher, sometimes if you want slices of salami, or you want slices of turkey or ham or whatever it might be, those slices are super thin. Well, this is even thinner than that. It's on a microscopic scale. And we're essentially looking down. So we're not looking longitudinally at the entire muscle. We're looking down inside the tubes. So think of them as just kind of sitting upwards. So you're looking down each of these muscle cells, excuse me. So here there's a different coloration of these muscles, and that's the key. That's the defining factor for how we can determine if it is a type 1 muscle cell or type 2 muscle cell. And I'll be explaining what that is again just a little bit. But the defining feature for the type 2 muscle cells, especially the type 2B or type 2X muscle cells, are that they're white. So if you do a stain like this, they look more white. And so between A and B, I'm already giving you the answer here. B is your type 2 muscle cells. You also have a slightly reddish version of it, which is a type 2A muscle fiber, not to be confused with my my letters here. Type 2A is a middle type of muscle fiber, which is a cross between the full version of the type 2, so type 2B/X, and the type 1. And that's why it's slightly reddish. However, the really dark colored muscle cell is a type 1 muscle cell, and that's because it contains a lot more myoglobin. It can attach to a lot more oxygen molecules. And we're going to see why actually in this lecture. So that's how we can determine this. It's called doing histology staining to get this kind of research or data. Now, to look at some of the differences, again, this is something you'd have to go back to the previous lecture, but I'm going to go over it shortly here, is that the type 2B or X, and I've eliminated the type 2A just because I want as much of a contrast between them as possible, because type 2A is right in the middle of these two cells, so that's how we're going to think about it. Type 2B or X cells have greater force generation and quicker force generation. So these two belong to the white cells, or the type 2B/X cells. But the greater fatigue resistance, so being able to continuously activate and not get tired, as well as having more mitochondria, are for the type 1 muscle cells. And here's a fun fact that I didn't actually tell my undergrads, but you might be interested in this, is that people who have diabetes and people who are obese tend to have higher levels of these muscle cells, type 2B/X, and have less of these type 1. Why might that be? Well, because you have lower mitochondria, or it's at least correlated, I should say, more mitochondria. And these are more fat-burning, quote-unquote fat-burning cells by comparison to these. And again, we'll go into that exactly why. So how do your muscles actually sustain activity? If you activate them, how do they actually sustain that ability? Well, we're going to go back to our cross-bridge cycling. Don't worry, you don't actually have to know all this. But just to walk you through, remember when calcium is released from the sarcoplasmic reticulum, it binds to this kind of reddish protein here, or orangish protein. And that orangish protein is called tropomyosin. And it's blocking the myosin head right here from actually interacting with actin. And the interaction between actin and myosin is what actually allows for the contraction of your muscles. So, and I would definitely encourage that you go back to that lecture, the first lecture, to see how that looks, to really get a more succinct view of, or more detailed view of what I'm talking about there. So calcium comes down, it binds to troponin, which is on tropomyosin, and then that shifts tropomyosin, and then the myosin head can then bind. So that's the situation we see here. that tryptomyosin has moved over, although it doesn't look like it by the diagram. And the myosin head is then bound to actin, which is this blue or purplish structure. And you have an ADP molecule and a phosphate molecule. And I think I told you in the last lecture, you don't need to remember those. But the point is, the big point here for step 1 is that myosin head will then bind to actin. In the second illustration, you have what's known as the power stroke. And this is something I did want you to remember, because here you have the release of ADP and phosphate. Again, not something you need to care about, but look at the orientation of the myosin head. It's from straight up to off to the side. And the reason for that is because it actually it's kind of like a rower. It has rowed itself, it has rowed, and now it needs to detach from the actin to then fulfill a new contraction. So here it has shifted over, and this will move the entire actin molecule over as well. So the next step is for ATP to then bind to release this myosin head from the actin molecule or the actin protein. So once ATP binds, and that's all that happens, it just binds, then the myosin head will be pulled off of the actin molecule protein. However, it's still in its power stroke formation, like it's spent. It can't stroke anymore. It can't power stroke anymore. So what needs to happen is a process known as ATP hydrolysis. And if you're familiar with any sort of cell biology, this is the life force of your body. For your cells need to have the ability to undergo ATP hydrolysis. So the way that they do that is by just using water. So they use water molecules, and they end up cutting ATP to the ADP that I was mentioning earlier, that's not that important, and phosphate. So that whole process has to occur trillions of times. So every time you're moving your muscles, every time you're moving your body, me talking, you listening, you moving your head around, whatever it is, running, anything, that process has to happen trillions of times, and it happens thousands and thousands of times within each muscle cell, and you have millions of muscle cells. So it's an incredible process that occurs. But how does that happen? How do we actually continue that whole process? Well, right, I did mention ATP, so we need to have energy. And without this energy, none of this occurs. And that's why you get what's known as rigor mortis. So when a person dies, they run out of ATP because their body's no longer generating ATP, and they get stuck in this state, right? Because there's no ATP to remove the myosin head from the actin, so it gets stuck in stage 2. So myosin is still bound to actin, and you get a really stiff body. It stiffens up. So that's why we need energy to be able to constantly move through this cycle known as cross-bridge cycling. Okay, so how does it happen? Let's look at some of the metabolism. Let's look at some of the cellular aspects of this. So here we've got our muscle cell, and we're zooming into the muscle cell, and we're looking at the sarcolemma, which is the cell membrane, another fancy term by muscle biologists. And here we've got our mitochondria. But do mitochondria actually look like this? They do not. I'm going to show you some real images of what mitochondria look like. And here it actually looks quite similar. So let me go back real quick. This is, relatively similar to this, right? You get, let me go back one more time. You get what's known as the cristae. So you have this kind of bean shape, and you have the cristae, which is the folds that you see here. Well, you see the bean shape, and you see the cristae that are all going throughout. So this looks a lot like those mitochondria. Now, this particular technique is known as electron microscopy. It is, I think, one of the coolest things I've ever seen in terms of its ability. So to think that we're not just looking at a cell here. We're looking at within a cell. We're looking at an organelle within a cell. And with an even more advanced version of electron microscopy, which is just a super, super powerful microscope, With that power, we can actually also visualize proteins. So we can visualize actin, we can visualize myosin, we can actually see these proteins. And that technique is known as cryo-EM, so cryo-electron microscopy. So, but do all mitochondria always look like this? No, actually they don't. And actually the mitochondria that I work with look more like this. This is through a different technique. This is known as fluorescent microscopy. And fluorescent microscopy allows us to kind of distinguish different parts of the cell from one another by their color. And that's exactly what's being done here. So the white are the mitochondria, and you can distinguish that from other sections of the cell. I don't know what the pink is. You'd have to know what the protein is that was, or what the antibody was, the targeting that they used to illuminate these. And then you get this image, and then you can take this image and then you can quantify it. But you can see that mitochondria are much longer, right? And this is obviously not as much of a magnification as with this. Magnification here is much higher than here. But still, you can tell that maybe there's one bean-shaped, 2, 3, 4, but you also have these longer mitochondria. And that is quite common in cells. So don't think that it's just like this. Okay, so with that little side tangent out of the way, let's go ahead and discuss metabolism in muscle. So you've got your blood glucose, your blood sugar levels. So same thing that people measure for diabetes, same thing that they measure for hypoglycemia. And it's outside of the cell. So this is the inside of the cell, the inside of the muscle cell. This is our cell membrane, also known as the sarcolemma. And then this is the outside of the cell. So outside of the cell is blood glucose. It enters the cell, and this is a really basic image, because technically you would see AKT activation, AS160, IRS1 activation, insulin receptor, all kinds of stuff, and GLUT translocation. The point is there's a lot of complexity here, but here, let's just pretend none of that exists. And let's just say that it just enters the cell, which it technically does. So blood glucose enters the cell and it goes through a bunch of metabolic processes all through this yellow line that I'm not showing you. But here's the kind of the big picture, is that without oxygen present or insufficient oxygen present, you get what's known as a low ATP yield. ATP is the molecule that our cells use to contract. It's also the molecule that we use for almost everything else, but let's just focus on contraction for the time being. You get a low ATP yield. You get about 2 ATP, maybe 4, depending on how much of the blood sugar, or how much of the sugar or blood glucose is coming from the blood as opposed to glycogen. If it's coming from glycogen, you get more ATP. If it's from blood sugar, then it comes from, then it's going to be just 2. So let's just go 2 ATP. Really, really low yield of ATP. And you'll see why. By comparison to if oxygen is present, let's say sufficient oxygen is present, then that means that it goes through the mitochondria, which is why I mentioned it earlier, and you get a moderate ATP yield. You get about 30 to 32 ATP. So a huge difference between low ATP of 2 to in the 30s of ATP. Huge, huge difference. Okay, so what's the highest yield that you can have? Well, the highest yield you can have is coming from fat molecules. So fat molecules end up going through the mitochondria, and they can lead to a high ATP yield of around 130 ATP. So much, much higher than the low ATP yield, and even much higher than the moderate ATP yield. And this will also vary based off of the type of fat that you're talking about. So a longer fat typically yields higher ATP, I think 140 ATP, something along those lines. And shorter fats will be more around 130. So there is some variability, but it's not huge in the grand scheme of things. The point is that fat molecules lead to the highest ATP yield. Okay, so that's how it works, right? So that's just a really basic explanation, but What are the two metabolic processes that actually allow this to occur? Well, for blood sugar, you should always be thinking glycolysis. That is the metabolic process that allows you to either go oxygen absent or oxygen present, regardless, it's going through glycolysis. So blood sugar gets transformed chemically to all these different molecules, like fructose, or it does get turned into fructose at one point. Glucose 6-phosphate, like there's pyruvate, like there's a series of different chemical reactions that come from sugar that a bunch of enzymes, essentially like factory workers, work on one molecule of sugar and turn it into another molecule. If that is lactate, which is the low ATP yield, or if it gets shuttled into the mitochondria as pyruvate and will then lead to moderate ATP yield. And if it goes into the mitochondrion, then you are going to undergo a process known as oxidative phosphorylation. Now, I want you to focus on this word oxidative. That is because it's always oxygen present. Oxidative phosphorylation does not occur without oxygen. That's why the oxygen absent is all the way over here and it's not encompassed here. So oxidative phosphorylation yields a high ATP yield depending on the substrate. So if it's fat, it's high ATP. If it's blood sugar, it's moderate ATP in the grand scheme of these three. Now, why would we ever want to go low ATP yield or high ATP yield? Well, there are trade-offs between the two. So glycolysis is a fast production of ATP. So while the yield is very low, it is produced at a much quicker rate than oxidative phosphorylation, which is dependent on oxygen and is slow production. So while the overall yield of ATP that you'll get is much higher, regardless of if it's blood sugar or fat, the overall speed that you get that ATP is much slower. And that is quite problematic, as we'll see. So this is kind of a summary here that blood sugar goes through glycolysis. It leads to low to moderate yield of ATP. It is a fast production unless it's oxygen dependent, which I probably should have added that to the slide, but fast production unless it's going through the mitochondrion, and it's short duration. And that's because you get that burning sensation when you're exercising, for example, but that will be explained as well in just a little bit. Fat molecules, on the other hand, go through oxidative phosphorylation exclusively, and they yield high ATP yield, but this trade-off is that they're slow production. So you could do long duration stuff with that because you have so much fat on your body, but you just can't do it very quickly. So that's important. All right, so I've talked about metabolism, I've talked about glycolysis, I've talked about oxidative phosphorylation, but you may be familiar with creatine. Creatine is actually, it's not just another supplement. It's just thrown out there. It is a supplement, but creatine actually is its own independent metabolic pathway. So I'm going to explain it in the classical sense, but creatine does a lot more than what I'm going to explain here. That's why I wrote a book on it, because I was so fascinated by it. Okay, so creatine, the molecule looks like this, but do we actually need to know the structure of the molecule? No, we don't. So I'm just going to eliminate that, and we'll just pretend this is what it looks like. And so we've got creatine in a creatine powder form. And what does it do? Well, it turns out that creatine will go inside the muscle cells, and it does something to that muscle cell. But within the muscle cell, there's already creatine. Your body, even if you never touch a creatine supplement the entirety of your life, your body's still producing creatine. It uses methionine, arginine, and I forgot the third amino acid, and it will interchange between the kidneys, and the liver will interchange molecules, and the liver will then spit out creatine molecules, which will then enter your bloodstream and enter the muscle cells. So then these muscle cells suddenly get an increase in creatine concentration. So what does creatine do? Well, what creatine does is it takes our ATP molecule, which I've kind of detailed a little bit more than just a yellow splotch or a yellow sun or explosion of energy. ATP is an adenosine molecule with 3 phosphate molecules. So you've got your terminal phosphate, you've got, I forget what this one's called, the beta phosphate and the alpha phosphate, something like that. Gamma, beta, alpha, something along those lines. But you have these 3 phosphates that are linked to this adenosine molecule. And that is adenosine triphosphate. That's why it's called ATP. So ATP will be used by the cell, and they will anchor one of these phosphates, the terminal phosphate, the gamma phosphate, onto a creatine molecule, and this will generate phosphocreatine. Very original name, but I appreciate the simplicity of it, that you have creatine, this is a phosphate, stick the two together, phosphocreatine. Great. Okay, so why does that matter? Why would we care? Like, who cares if the cell does that? Well, a cell doesn't do that for no reason. It has to take care of low cellular energy. So if there are ever moments or times where the energy state of the cell is low, then what happens, and it's defined as low when there's an increase in ADP, so adenosine diphosphate, which is, as you can see here by my diagram, there's only 2 phosphates as opposed to the original 3. So this is a low energy state. But then you've got a bunch of phosphocreatine around. So what happens is ADP will interact with phosphocreatine and regenerate ATP. So when you are not in an when you have plenty of energy around, you create a reserve of phosphocreatine. So you kind of pay your dues up front, and then when you need them, then this is when you need them, exercise or you're running away from a bear, which happens all too often. And then this phosphocreatine will then interact with ADP to regenerate ATP, so you can recycle it really quickly, super, super quick. So that is why this system is the fastest metabolic system, even faster than glycolysis. So what's the big thing here? Well, they're extremely quick energy source. That's one thing. And secondly, only lasts about 10 to 15 seconds. So you don't have a whole lot. And the more you supplement, the longer this will last. But it's not going to last minutes or anything like that, even if you supplement with kilograms of creatine. And it is independent of glucose and fat, because you didn't see anything about glucose and fat when I mentioned it. And that's because it is completely independent. But it's only going to last you about 15 seconds at most. So in that situation, then you have to switch to glucose or blood sugar or sugar in general and fat. Okay, so let me ask you, based off of the information that you have here, if you're watching live or if you're watching after the fact, what energy source are these people using? So if they're weightlifting, what energy source would they be using? Would they be using phosphocreatine? Would they be using glycolysis? Would they be using fat or oxidative phosphorylation? Or would they be using a combination of them, and which ones would they be using? What about marathon running? Remember what I said, that there's a trade-off. Some of them are slow in terms of producing ATP but can generate a lot, and other ones are quick but don't generate very much. So I'm going to give you the answer. So pause the video or pause the podcast if you want to think about it some more. But the correct answer is that weightlifting will use phosphocreatine and glycolysis. On the other hand, marathon running will use glycolysis and oxidative phosphorylation, and that's because you need a constant supply. And it's the aim when you're marathon running, anytime you're doing any long distance exercise, the aim is to try to be as metabolically flexible as possible so that your body cells will use more fat than they will rely on glucose or carbohydrates. Because if you run out of carbohydrates, you end up going through what's known as bonking or hitting the wall, because then you only have your fat reserves. And as I said, the production of energy is super slow for that. So there you go. There's the answer. Okay, so now returning back to our muscle types, and here I've got them all 3 listed. So type 2B/X, which are fast twitch, also known as glycolytic, also known as white muscle cells. And then type 2A, which is also fast twitch, but they're kind of a combination of oxidative and glycolytic, and they're kind of reddish muscle cells. And then you've got your type 1, which are slow twitch, oxidative completely, and red muscle cells. So what does that mean in a metabolism sense? Well, we're not focusing on the contraction. We're not focusing on the color. We're focused on what the source of energy is and what kind of metabolism is used. So type 2B/X is going to use glucose. It's going to use glycolysis. Type 2A, which is oxidative glycolytic, is going to use both. And this is a pretty rudimentary way of understanding this, because really all these have the ability to use any style. It's just that they focus more on one or the other. Type 2A are really plastic because they can switch more towards the type 2B or more towards the type 1, depending on how you train them. So they have the capacity for both. And then finally, type 1 are glucose and fat centric, so they use both, but they always, or not always, but they typically go through oxidative phosphorylation. So they're not going to end up using glycolysis as much. Well, they'll use glycolysis, but not the oxygen-independent mechanism. Okay, so here I wanted to finish this off with a mystery. So imagine you and I we are the experts. So I have educated you. You are now an expert in muscle physiology. And based on just the information that I gave you, so if your mind starts wandering to other possibilities, just don't. Because I've given you the answers, but I'm going to walk you through it. So if we have time, here we go. Here's the situation. A teenage boy is brought to the emergency room, and the doctor on duty immediately knows the situation is serious. So she has him sent to a hospital. At the hospital, a team of residents run tests on the kid and find that his tests come back normal, except for elevated white blood cells. That is a clue right there that you don't need to know, but I'm just going to point it out. For my class, they should have known that that was a clue when I was going through it. So extremely confused, the doctors at the hospital, call upon a room of experts. That's you. To figure out what is wrong with the patient. You must find out what is wrong before his condition worsens beyond saving. All right, so let's look at some clinical stuff, and then we're going to go into some experiments that are going to give us some more clues. The first thing is, here's an image of what's going on here. And his symptoms are that he's paralyzed. He can't open his eyes. He has difficulty breathing, so it's labored breathing, and it's becoming even more labored as time is progressing. And he has facial weakness, so he can't smile. Beyond just being paralyzed overall for his whole body, he also has facial weakness. So can't move his mouth, can't talk very well. Okay, so you request more blood tests, and you end up looking first for his blood work. And you find in his blood work, if we actually took a sample of his blood and put it under a microscope, this is what we might see. And normally, under normal circumstances, I'll tell you that this is not what you're supposed to find. You're not supposed to find these are bacteria. So you're not supposed to find a series of bacteria. And that is not the answer to the riddle, just so you know, but it's certainly a component of the answer. So we've got bacteria in his blood, so keep that in mind. So this is all under the microscope. I've kind of zoomed in here. Okay, so muscle biopsy. Based off of what we know from even earlier in this lecture, this is a different type, different stain, but between the 2 or 3 different muscle types that I offered before, is that we had the type 1, the type 2A, and type 2B, or type 2X, but we're just going to call it type 2B. And here we only have 2 colors. So let's just separate it out as the 2 different types of muscle cells that we have. So we've got type 2, and type 1. Which ones are type 1 and which ones are type 2 based off of the knowledge that we have up to this point. So again, pause the video or pause the podcast if you want to take a guess. take a gander, and I'm going to give you the answer now. So again, just based off of this test, we have the red fibers, right, which are more oxidative, which are more filled with myoglobin, which attach to more oxygen, have more mitochondria. Those are the type 1 cells. And the lighter color is the type 2 cells. So keep that in mind. So is there anything abnormal about this? Well, to the untrained eye, you might see this white patch here and you might think, oh, maybe something's wrong there. But I didn't teach you anything about that. So just assume that that's fine. And this is actually just collagen. That's all it is. It's just matrix protein. It's not anything related to the muscle cells, and it's perfectly healthy. So this image is actually perfectly healthy. There's nothing abnormal here, except for maybe that there's more type 2 cells than there are type 1 cells. That's just about it. So another question we have to ask ourselves is, does it contract when we artificially stimulate it? So we artificially stimulate the musculature. Does it contract? Do we get the myosin and actin binding to one another and leading to a shortening of the muscle cells? And I'm just going to go ahead and tell you that based off of this test, the answer is yes. So up to now, we know that this person is paralyzed. They have a hard time speaking. They have facial weakness, have trouble breathing. So those are the clinical aspects. Then we've run a few tests so far. We've tested their blood and we know that there's bacteria. So that's wrong. So that's something that's wrong, but we need to figure out exactly what that bacteria how that might be implicated. We looked at the muscle. We took a muscle biopsy and we plated them and we looked at them and we saw that sure, there's, type 1 muscle cells, there's type 2 muscle cells, but nothing jumps out at us. And then when we actually contract the musculature, we see that it does contract normally. Okay. So let's keep investigating. So now we're going to look at our nerves. So look at his nerves. So let's say, not that you can actually technically do this unless you're looking at like animals, but let's say you were to take a sample of nerve, or you were to just open him up somehow and test his nerves. So here we've got an actual image of a nerve or neuron. And here, let's say that this is the axon. So this is the cell body, and the axon is leading down to a muscle cell. I'm going to show you a really cool image of that in just a second. But here we've got our image, and we do a test. So we're going to probe this axon and see if there is what? What would we probe for on a neuron? I only taught you about one thing. And that one thing is the action potential. So we probe this neuron and we see this come up. Does this look normal? At the very beginning of this podcast, of this lecture, I taught you just really brief understanding of action potentials. So if you were to look at this, and even if you just go back right now, pause it, go back and just look at the picture and compare, if you want to do it that simply, does this look normal? So again, you've got your negative membrane potential inside the neuron in this situation, and then you have an action potential that goes up, and then you have repolarization. It says depolarization, repolarization, hyperpolarization, and then back to normal. Is that normal? Okay, so I'll tell you the answer. The answer is yes, this is normal. So far, we have only found one thing wrong with this kid. Only one thing has been wrong with this kid. So now let's get even more granular here. Let's start to look, and I'm going to ask you a question here that's not really planned, but I'm curious if people that watched the previous podcast can identify this. So here we've got our neuron. We've got our axon that's coming down and it branches, and here we've got our axon terminals. What are these green striations here? And I kind of gave you a clue if you watched the first podcast. The word striation should give you an indication of what's going on here. So what is this? What is this whole structure here? I told you that this is the neuron. And what is this? Okay. Hopefully I gave you enough of a pause. The answer is that these are muscle cells. So these are muscle cells, and the red that you're seeing here this is again fluorescent work, just like I showed you at the mitochondria. This is we're using fluorescent work here to actually fluoresce proteins on each one of these structures. So here they've done green, maybe they did actin, I don't know, something along those lines. Something that's both in neurons and in muscles, so they're both showing up as green. And this red here, really cool. If you go back again to the very beginning of this podcast, I told you about acetylcholine receptors, and that's what they've got fluorescing red. So you're actually seeing right now some fluorescing acetylcholine receptors, fluorescing neuron, and fluorescing muscle cell. Pretty damn cool, I got to say. Okay, but so now we're going to zoom in. We're going to zoom into this and look at what's going on. And remember this, I've kind of color-coded things a little bit differently here. So we've got our action potential that's coming in, and let's say, to direct you, let's say that the action potential has occurred. It's gone through the entire neuron and it is now over. Okay, so remember the action potential comes down. This action potential right here comes down the neuron and reaches the end. And what is supposed to happen? Does this look normal? You have to ask yourself again, does this look normal? I'm going to pause for a second, but just think about what happened at the beginning of this podcast when I discussed acetylcholine, its relationship to action potentials, and et cetera, et cetera. So the answer is no, this is not normal. So this is abnormal in that, and hopefully you can guess this already, that acetylcholine was supposed to bind to the cell membrane, the neuron cell membrane, and release its acetylcholine. But for some reason it didn't. So can you guess the relationship between the bacteria and this? What are the bacteria affecting? Well, I'll tell you the details of the information here. And I still have one more question for you, which I'll ask you as doctors, as experts, what you would do to cure this particular condition. And there are a number of different answers. So don't, post in the comments what you think you would do to solve this problem after I explain it. So in a normal situation, what you have is a synaptic vesicle, which contains the acetylcholine, and clearly I did make this diagram, so the colors are all different. And on that synaptic vesicle, you have what's known as snap and snare proteins. So snare proteins and snap proteins. The snap proteins are found on the wall on the cell membrane, the inside of the cell membrane of neurons. And those will literally entangle themselves or interact with snare proteins on the synaptic vesicle. Once that happens, which is right here, this then allows these proteins to kind of pull in the vesicle, which then binds to the cell membrane and releases acetylcholine. Acetylcholine will then bind to the acetylcholine receptors and allow for sodium and the action potential to propagate to the muscle cell. This is what is normally supposed to happen. This is what we covered earlier, except for the snare proteins. On the other hand, however, here, from this bacteria, which is known as Clostridium botulinum, hopefully I'm pronouncing that correctly, but it is botulism. So I don't usually look at the live chat too much, but Joseph, yes, you got it right. You're exactly right. It is botulism poisoning. So the bacteria itself may not actually be detrimental. However, what they are releasing is detrimental in that they release a toxin. That toxin, which is a protein, interacts with receptors on the cell surface, the neuron cell surface, and gets what's known as endocytosed or taken up by the cell. Once it is inside the cell, it is released inside the cell, right here, and then it will cut up which is known as a protease. It will cut up the snare proteins as well as the SNAP proteins. So SNAP proteins and the snare proteins. So without these proteins, then the vesicles no longer have an ability to dock to the cell membrane and can therefore no longer be released into the synaptic cleft to bind to the acetylcholine receptors. So that's what's going on. That is the issue. So we're not getting transmission from the neuron to the muscle cells, and therefore you're getting, as it builds up, as you get more and more of this botulism toxin, it's affecting more and more of the nerves, and therefore, if we don't do anything, this kid's going to die. So what can we do? That is my final question to you. What kind of treatments would you recommend to the hospital to save the boy's life? And you can get pretty creative with this. There are a number of different things that you could try. So I'll pause here just for a second, and I just want you to think about the different ways. So again, you've got bacteria that are flooding into a system, that are in a system, and then they are releasing this toxin, which the toxin itself is the real issue that's affecting the neurons. So how would you deal with this? And there are a number of different remedies. So let's walk through a few of them. And you may have more than the ones that I'm going to mention now. So one of them is super simple. Antibiotic. Done. Boom. We just nailed it. You just hit an antibiotic, it kills the bacteria, and eventually the toxin will be removed from the body. However, let's assume that things have gotten so bad that an antibiotic is not going to be enough. So in this situation, what is something else you could do? Well, another thing you could do is throw in if you've ever heard of the term chelation. So when people talk about actually, let me cut so I can actually look at you guys. When people think about like removing toxins from the body, have you ever heard somebody say, "Oh, you got to take my detoxifying supplement"? Except when I'm joking. Sometimes I throw it up in my videos of like, "Buy my detox supplement" and all that. A long time ago, I did some reading on detoxes and stuff like that, and really detoxes don't do jack, but however, in really toxic situations, like I'm talking true toxic situations, I'm talking like you go through and you like you bathe in some sort of substance that leads to a toxin that enters your body, like real biohazard kind of stuff. In that situation, then there are chelating agents. So there are things that doctors will inject or give you to consume, and those will then bind to the toxin and eliminate it. So once it's bound, the toxin can't do anything because it's stuck to that chelating agent. So in this situation, what we can do is a similar thing, that we can inject the kid with something that will bind to the botulism toxin, and then it can't interact with that receptor. So let me switch back to this real quick. So remember, it binds to this receptor and enters the neuron this way. So if we can block this, then it can no longer interact, even though this still exists. So that is another way that we could deal with it. Another way would be to use both treatments at the same time. Another one would be to maybe use some sort of I remember when I was teaching this, actually, somebody threw out a bacteriophage, which I found hilarious. If you know what a bacteriophage is, it's essentially a virus. It's quote unquote a virus that affects bacteria. So if you want to get really creative, like if you're super desperate, you could release a virus that specifically affects bacteria. And then, those bacteria are going to have a tough time to survive. So that's another possibility. Another possibility would be to use a protease or a cleaving chemical that cleaves the toxin itself. So to find one that's actually specific enough to do that would be really tough, but we're getting creative here. So you release that into the bloodstream and it starts cutting up the toxin specifically, and therefore then you just have pieces of toxin that end up getting cleared. out of your system. So the point is, there's a lot of different things that you can do to try to help this young man. All right. Well, hopefully you found this informative. Hopefully you had fun with this. I try to piece together. I think I like the idea of kind of creating riddles every once in a while, as long as I can kind of set it up correctly with enough information so people can kind of guess this stuff, which Joseph on the live chat freaking killed it. Guessed everything. And as he pointed out, something I guess I should also point out is botulism poisoning is the same thing that people use for Botox so they inject it, but it's local, so that it's one, you're not injecting the bacteria, you're injecting the toxin itself. But on top of that, it's local, so it's not going to be going systemically across your body, because the problem is it starts affecting your lungs, your diaphragm, so that you can't breathe and then you end up dying. So it is a time-sensitive issue. So anyway, hopefully you enjoyed this. I hope you did. If you did, share it. Comment below, like, do all the good stuff, subscribe. And with that, I hope to have the absolute pleasure of speaking with you in the next one. Have a good one. Bye.